Welcome, everyone, and thank you for joining the webinar. We're going to wait just another minute or two as we still have folks who are joining. So we'll get started in just another minute. Thanks for being here. All right. Thank you, everyone, for joining us today. My name is Patrick Sullivan. I'm at the Rollins School of Public Health at Emory University, and I'm happy to co-host today with Dr. Ken Mayer from Fenway and Harvard. And we have a really exciting set of talks for you today on the theme of the future of PrEP. So I'm going to briefly introduce our three speakers. We'll have three talks, which will go back to back, and then we'll have a time for discussion at the end. I encourage you, if questions come up during the talks, feel free to use the chat feature to post your question. And after the talks are done, Dr. Mayer will moderate a discussion using the questions that you've posted. And if you'd rather wait and put your virtual hand up at the end, you can do that too, and we'll have some time for discussion. So today we have three fantastic speakers. Dr. Maggie Charnogorski is the head of digital innovation and implementation science at ViiV Healthcare. Before coming to ViiV, she served as the deputy director of Comprehensive Women's Health at the Department of Veterans Affairs. From 2014 to 2015, Maggie served as senior policy advisor and senior medical advisor at the Office of National AIDS Policy in the White House under the Obama administration. From 2019 to 2013, Maggie served as deputy director, HIV, hepatitis, and public health pathogens at the VA. Maggie's a practicing HIV infectious disease physician, has a weekly clinic at the DC VA Medical Center. She received her MD from Georgetown University School of Medicine and completed internal medicine residency and ID fellowship at George Washington University, where she also received a master's degree in public health. Welcome. After Maggie will be Dr. Mopali Das, who is Vice President of Clinical Development, HIV Prevention, and Virology Pediatrics at Gilead Sciences. As head of HIV prevention, she leads high-performing cross-functional teams accountable for the overall development of strategy and the conception, design, and execution of HIV prevention clinical trials from phase 1 through phase 4. She's dedicated to ensuring efficient development and regulatory approvals to support rapid access to innovative products to help end the HIV epidemic. She also oversees the pediatrics team, who are deeply committed to ensuring the development of pediatric formulations for Gilead's virology medications. And our third speaker will be Dr. Rebecca Plank, who's an infectious disease physician who's dedicated her career to HIV treatment and prevention research. Before joining Merck in 2015, Rebecca was a full-time faculty member at the Brigham and Women's Hospital and at the Harvard T.H. Chan School of Public Health in Boston. She received her medical degree from the University of California, San Francisco, and her MPH from the Harvard T.H. Chan School of Public Health. She completed her residency and a chief resident year in internal medicine at Brigham and Women's Hospital. Rebecca was an ID fellow at the combined Mass General Brigham and Women's Hospital program and the HIV fellow at Brigham and Women's. She spent several years working in HIV treatment and prevention research and providing clinical care with the Botswana-Harvard partnership and later with the Nyanza Reproductive Health Society in Kenya. At Merck, Rebecca is a senior principal scientist and product development team lead for the MK8527 oral once monthly PrEP program. So I think you can see we have a distinguished group of speakers and an exciting topic. So without further ado, I'll turn it over to Dr. Charnogorski.
Thank you, Patrick, and thank you, Ken, for inviting me today with this distinguished panel to present for you today. Hold on one second. Let me share my slides. So hello, everybody. Once again, my name is Maggie Chernogorski. I'm the head of Digital Innovation and Implementation Science at ViiV Healthcare. And I'm here today to talk to you about the implementation, excuse me, science portfolio of studies for CAB-LA for PrEP. So as many of you know, CAB-LA for PrEP is the only currently approved long-acting option for PrEP. It's dosed 6 times a year and demonstrated superior efficacy over daily oral TDF and FTC in 2 kind of pivotal clinical trials, HPTN 083 and HPTN 084. For today's presentation, I'm actually not going to go over the clinical data, but if you guys are not familiar with it, I'm happy to send some materials after this call. But what I want to focus on today is the implementation science data, which is data that's complementary to the efficacy data. Now, as we all know, long-acting PrEP is a paradigm shift in the way healthcare delivery is currently administered to patients. And although we are very adaptable in the HIV space, change doesn't happen overnight. So what we wanted to do at ViiV is to kind of complement the efficacy data with an implementation science strategy to help us kind of achieve 2 key goals. One is to expand PrEP choice and uptake in all patients who can benefit from PrEP. And 2 is to potentially accelerate the adoption curve for PrEP in these populations 3, 4, 5 times faster than what we saw with oral PrEP. Many of us on the call probably understand and believe that we want to end the HIV epidemic. And to do so, there's a couple of key things that we need to do, right? On the treatment side, we need to make sure that everybody is diagnosed, on therapy, and virally suppressed. But on the PrEP side, we know that currently oral therapy has only reached 20, 30%, 35%, depending on the context, of people who can benefit from PrEP. And if we really want to get to the end of the epidemic, we really need to expand PrEP options and improve access to all people who can benefit from PrEP. And hopefully we can do this faster than what we've seen with oral PrEP. Oral PrEP data has been available since 2011. And although we've seen dramatic increases over the last couple of years, we're still only reaching about 20 to 30, 35% of people. So to expand the PrEP services, we wanted to look at kind of this strategic approach to thinking about implementation that we call the layer plus approach. So what that means for us in the layer circle is we know that PrEP is now currently being offered to a percentage of people in the world that could benefit from PrEP. And how can we best integrate CAB-LA into current PrEP delivery models? But like I said, PrEP is currently only reaching a certain percentage of patients. So what we wanted to look at is also beyond just layering CAB-LA into existing PrEP models. How do we expand CAB for long-acting for PrEP into new contexts and new populations? And as you can see here on the left, that's actually the bigger part of the circle because we still have quite a bit of people to reach. And to do this, we kind of identified some key implementation gaps that we wanted to address. Everything from service delivery models to testing to provider support to understanding the populations and how they may access or benefit from PrEP. What does demand creation look like? What does choice and switch look like? How are outcomes along the PrEP cascade? So not just initiation and adherence, but how do we help promote retention and persistence on CAB-LA and PrEP in general, and looking at costs. And all of this, we created kind of an implementation science portfolio to strategically address all of these questions. And we have a number of studies, both in the supported, collaborative, and investigator-sponsored space to address this. A lot of these studies are still ongoing. I'm going to kind of talk through some of the data that's been presented to date right now, and then talk about how we're going to move forward in the future. So the first study I want to talk about is our PILLAR study, which was kind of our first US study focused on that layer approach, where we wanted to evaluate integrating CAB-LA into PrEP into standard of care for existing PrEP sites. So these are sites that are currently delivering PrEP services. In this study, we focused on MSM and transgender men. And I just want to pause for a second and focus on the populations and how they were gender aligned. We did a lot of community feedback and community assessment of how to design the study. And we heard loud and clear that transgender men wanted to be in a gender aligned study. So Pillar was actually the first US study that had that approach. And we're really proud that this is how we are designing all our studies moving forward. In this study, we had two arms for implementation. One was the dynamic implementation arm, and the other was the routine implementation arm. The dynamic arm included interventions like peer-to-peer best practice sharing, CQI, telehealth, and a lot of different kind of digital support tools, while the routine arm was asked to implement CAB as they would any other PrEP service. And we actually learned quite a bit from both arms along the way. We used CAB Q2 monthly in the study, and it was a hybrid 3 study. So we looked at both clinical outcomes as well as implementation outcomes in this study. As much as we strategically chose the patients that we wanted to participate in the study that could benefit from PrEP, we also wanted to choose the clinics pretty strategically. So we looked at high prevalence areas, but in those high prevalence areas, we know that not all clinics are designed the same. They don't all have the same resources. They don't have all the same kind of staffing, and they're not set up exactly the same. So we strategically chose different kinds of clinics to participate in this study. Everything from federally qualified health centers, community-based clinics, to private clinics, academic institutions, and integrated healthcare systems like AHF. In terms of patient population, I already talked about this, but we really wanted to follow the epidemiology. And we actually exceeded our diversity targets in the study with over 60% Black and Latino participants and 6% transgender men, which followed the epidemiology. At the most recent CROI conference, we presented some clinical results, and I'm not going to spend too much time on this, but it was really great to see that not only are we seeing high persistence on CAB-LA through month 12 in this study, but we're seeing no acquisitions, HIV seroconversions through month 12 in the participants in the study. And we also saw a high fidelity to the plus or minus 7-day injection window through month 12, showing consistency and great clinical results in this study. But what I want to focus on a little bit is on the implementation results. What we found was that we wanted to focus on kind of key 6 areas for implementation. This included everything from practice preparation to patient selection to benefits verification, inventory management, kind of scheduling and rescheduling injection visits, and continuation and persistence. And what we found was actually there's more similarities than differences across these clinic types, which was interesting for us to understand. The dynamic arm did reach kind of steady state implementation or felt that they integrated CAB-LA into routine care a little bit more quickly than the routine arms. On average, it took them about 3 months. But the routine arms also felt like they confidently integrated CAB-LA into their routine clinical practices in about 6 months. So although this wasn't overnight, we know that it can be done, and it doesn't take extremely long to integrate if you have the right tools and strategies in place. The support tools such as schedulers and trackers, digital support tools for inventory management and acquisition were really helpful in this study, as was telehealth for helping support persistence and continuation. And although it wasn't a one-size-fits-all approach, what we were able to generate was kind of a menu or a suite of options to help support clinics, regardless of what kind of setting you were in and what initial resources and structure you had set up. With some of these strategies and tools, we were able to help support all of these clinics to integrate CAB-LA pretty quickly. And peer-to-peer best practice sharing in the dynamic arm was hugely valuable, and it's something that we're looking to kind of integrate moving forward and really amplify to help support newer clinics who are looking to integrate PrEP into their systems, or CAB-LA for PrEP into their systems. Now, the Ebony study is kind of our sister study to Pillar. And this study was designed to kind of look at that plus part of the circle in the strategy. In this study, we wanted to evaluate CAB-LA for PrEP for standard of care for Black, cis, and transgender women who are receiving routine care in the US. Now, we know that HIV negative Black women are often not going to PrEP service delivery clinics currently. They're not seeking out the services of HIV clinics. They're getting their care in centers like family practice or women's health or OBGYN services, transgender clinics. And so what we wanted to understand is what are the implementation strategies to help support this population and these clinics to integrate CAB-LA into routine care. In this study, we had 3 different arms: the standard implementation arm, the enhanced implementation arm, and the enhanced collaborative arm. We used Q2 monthly CAB-LA injections. And once again, it was a hybrid 3 trial. For those of you that know implementation science, where we looked at clinical outcomes and both implementation outcomes. And what we found here was that, so the enrollment characteristics here were also similar to what we did in the PILLAR study. We looked at high prevalence settings, but once again, we kind of carefully chose the clinics that we wanted to participate in this study, looking at those women's health clinics, OBGYN, family practice, transgender clinics, because we wanted to understand what the needs were from the clinic perspective, regardless of how they were initially set up. We enrolled 163 participants, and we achieved our target enrollment of 20 to 30% transgender participants. So this study is still ongoing, and so we don't have all of the results yet, but some of the things that I can share with you today was that amongst the patient population, there were some similarities and differences amongst transgender women and cisgender women. The transgender community had more knowledge and awareness of PrEP in general, and there was more of a positive community attitude towards PrEP, whereas cisgender women were more likely to be PrEP naive. But both of these populations really wanted the most convenient option for PrEP for themselves, and they often wanted something that didn't require daily oral pills. And what we saw, or what we're seeing with the implementation here, is that these clinics actually were set up a little bit better initially to be able to implement long-acting injectables in their clinic. Many of these clinics already had familiarity with other kinds of injectables, and they were able to integrate kind of the Q2 monthly injections more readily into their workflow. They had more experience with specialty pharmacies and how to work with SOPs and billing codes and things like that. Where there needed to be a little bit more strategic kind of tools to be given is around addressing how to identify the best candidates for CAB-LA amongst this population, as well as helping support the providers and having discussions with their patients about PrEP. It wasn't something that providers in this community were doing regularly, and they needed some language to be able to offer CAB-LA in a non-stigmatizing way. We did a lot of role play. We created some scripts. We helped support them in integrating this into their routine conversations with their patients. Similarly, there were some digital tools that were extremely helpful, like the sexual health assessment, which is a digital tool that we provided to help identify patients and help them understand what their sexual health goals were and how PrEP can help achieve them. But between these 2 studies, I think we've started to develop quite an extensive menu, or a kind of a suite of options to help integrate CAB-LA, regardless if you are a current PrEP delivery center that is used to giving oral PrEP, what modifications and changes needed to happen to be able to integrate long-acting, as well as for clinics that weren't used to giving CAB-LA, what are the strategies that can help support integrating this into routine care for all different sites. And as we develop more and more of these strategies, we want to share best practices across new clinics that are coming on board. Another key area of interest for us, because there is a high need and a high demand, is in the low and middle income country settings. Here, there's a lot of opportunity to expand PrEP to patients that can benefit, and there's a lot of need. What we did here is we've kind of strategically chosen 9 implementation studies in 14 countries, and we've enrolled over 7,000 participants in what I consider kind of our 9 pivotal studies. These are collaborative studies that we're doing with our amazing global and regional partners like UNAIDS, USAID, PEPFAR, Bill and Melinda Gates Foundation. And as you can see, many of these studies are in various parts of the world with large sample sizes, reaching populations such as men, women, adolescents, and transgender communities. But what I want to focus your attention on is the fact that we're looking at lots of different delivery models, right? And I think there's a lot of learnings that we can get from these studies that we can apply globally. So we're looking at everything from private pharmacies to nurse-led community-based interventions to mobile health clinics to integration into primary care and sexual and reproductive health centers. One key study that I want to kind of highlight that has read out already in this region is the SEARCH DCP study, or the Dynamic Choice Prevention Study, led by Diane Havlier and her group out of UCSF in Uganda and Kenya. And in this study, they enrolled about 1,000 participants who could benefit from PrEP and gave them the option of oral PrEP, PEP, or CAB-LA, and gave them the option to either switch or stop based on their personal preferences and risk. The key endpoints here were coverage, feasibility, acceptability of PrEP, and HIV incidence. And I think what is really important to see in this study is that providing CAB-LA as a choice really increased the coverage of the number of people choosing to be on PrEP. And that's exactly what we need to do if we want to get to ending the epidemic, right? We need to expand the number of people who are willing and interested in using PrEP. The addition of CAB-LA into the DCP arm of the study significantly increased PrEP coverage across all the subgroups. 56% of the participants in the DCP arm chose CAB-LA, and 42% of them had never used PrEP or PEP before, showing that although oral PrEP is a great option for some people, there's a huge percentage of people that are not interested necessarily in using oral PrEP currently, and that by adding a long-acting option, we can really expand that pie of people using PrEP and really get us towards that ending the epidemic goal. Importantly, no participants in the study that received CAB-LA acquired HIV infection. And I mentioned the other studies just a moment ago, but a lot of them are still ongoing, but the insights from them are really similar and consistent across all the studies. We're seeing a high preference of CAB-LA in these cohorts. And importantly, we're reaching so many people who have never used PrEP before. And once again, to me, that is one of the key highlights coming out of these studies. We're seeing high persistence and 0 new infections in these studies. And so in summary, we have really great efficacy data that I know I didn't present today, but what we're seeing is really positive findings from these implementation science studies. And CAB-LA is performing even better in the real world than we've seen in clinical trials. What it's helping us do is helping us show that we can have high persistence on PrEP medications. CAB-LA is showing really high persistence, high coverage. Once again, importantly, expanding the number of new people on PrEP and showing strong effectiveness. And through these studies, we're hoping to kind of grow that portfolio of strategies to help support clinics, whether you're delivering, care in an existing PrEP clinic in California, or trying to reach women in Uganda or adolescents in Brazil, that we can help you have the strategies and support tools that can help you implement across diverse clinical settings and support the uptake of this really important medication today and faster than what we've seen with oral PrEP in the past. So what's next for long-acting cabotegravir? Well, last year at CROI in 2004, we presented some preliminary data that CAB-ULA exhibits, or ultra long-acting exhibits, a PK profile that supports 3 times a year dosing. And we're really excited about this data. We're seeing good absorption, longer half-lives, and we saw that CAB-ULA IM was better tolerated than sub-Q. So we're moving forward with the CAB-ULA IM injection for 3 times a year dosing. As we go forward with these studies, we're really hoping to see the ability to pull through some of the IS strategies that we've developed with the Q2 monthly IM to accelerate the uptake and the expansion into these new populations, into routine care, hopefully, even faster, 5-10 times faster than we've even seen for CAB2 monthly. So with that, I think that is my time for today. I'm happy to take questions at the end of this session. So thank you for your time today.
Great. Thanks, Maggie. Should I go ahead and share my slides, Ken?
Yes, please.
Yes.
Okay. Okay, can you guys see my slides?
Yes, perfect.
Is the little you're sharing your slides thing coming up? Okay. All right.
We don't see it. No.
Okay, great. Thanks. Very grateful to the organizers for the opportunity to share our prevention and clinical development program and give you guys some updates. Okay, so sorry, I think I'm constitutionally obligated to start with this slide, but here we go. I just want to highlight the timeline here. Between Atripla and Biktarvy, all drugs we know well from HIV treatment, is between 2006 and 2018. And that's relevant because that shows you exactly how long we've been working on lenacapavir. So we first started the capsid project in 2006. And it took a long time to get to the formulation that went first in human in 2018. And that was during a period of time of robust development by all the people on this call of other options for HIV treatment and prevention. But our brilliant medicinal chemists and scientists and the research group did not give up. And they, even after the first gen leads panned out and they'd already screened thousands of molecules, they got to the eventual clinical candidate of lenacapavir, which is really a unique molecule. And over the past several years, we've had the results for lenacapavir, along with other medications for the treatment of highly treatment experienced people with HIV, with multi-drug resistant HIV, as well as the results of the PURPOSE trials that I'll briefly overview and highlight some of the new information we know from the programs. So lenacapavir is the foundation of our long-acting portfolio, both for treatment and prevention. It is, as I mentioned, a unique molecule because of its very high potency, high stability, and that it works in multiple different steps in the viral replication cycle. It has no overlapping resistance with existing HIV meds and a very well understood efficacy and safety profile. And the unique nature of the molecule allows flexible dosing for oral as well as injectable through different parenteral formulations. Obviously my colleagues and I are dedicated and committed to developing new options for HIV treatment and prevention. So currently, obviously, we have daily treatment options and daily PrEP options. There is the long-acting every other month cabotegravir option for prevention, but we really want to get to twice yearly and once yearly, and potentially a longer acting oral option, either monthly or weekly. Okay, so with respect to the PURPOSE program, this is our tree slide where we like to talk about the entire set of programs. So PURPOSE 1, PURPOSE 2 were our pivotal trials that demonstrated safety and efficacy for lenacapavir for HIV prevention. Those trials are currently ongoing, and I'll tell you about the status of them. In addition, we have 3 other trials, PURPOSE 3, 4, and 5. PURPOSE 3 and 4 are collaborations with the HIV Prevention Trials Network, NIH, NIAID, and NIDA. Where PURPOSE 3 is focusing on US cisgender women because they could not have been included in the PURPOSE 1 trial due to the nature of requirements for the counterfactual design. PURPOSE 4 focuses on US people who inject drugs. I know we have a lot of our investigators on the call today, so I just want to thank all of you for working so hard on our program for lenacapavir for HIV prevention. And then in France and the UK, we have PURPOSE 5, which evaluates a novel endpoint. It's not to demonstrate safety or efficacy, but really to look at persistence over time and evaluate the potential advantage of a twice-yearly injection for persistence or adherence over a 1-year time period compared to daily oral PrEP. And we couldn't have done any of this program without our focus on the 4 pillars of health equity partnerships, including the voice of people who would benefit from PrEP and the community throughout our trials, setting up trial-specific global community advisory groups and seeking insights in partnership with our global stakeholders, a focus on person-centric design to lower the barrier for entry in randomized clinical trials, and a focus on health equity, including diversity, equity, and inclusion. And all of the trials were able to happen because of the proof of concept from HIV treatment, as well as a demonstrated safety and efficacy in treatment and proof of concept in the non-human primate studies that showed efficacy of lenacapavir and capsid inhibitors for preventing SHIV in male and female non-human primates. Okay, so I like to start with this slide just to remind us that we've been working on this since December of 2019, where we held our first stakeholder meeting in Kigali, Rwanda. And we heard many things about how we should do this set of trials differently that we were able to implement. And one of the things that we heard was nothing about us without us. And there were very strong voices encouraging us to include adolescents and people who could get pregnant in our clinical trials to make sure we had data on these 2 critical key populations at the time of the data in adult populations, and that is what we did. So starting in 2019 and continuing on to 2025, we have been meeting regularly with our stakeholders, including in broad stakeholder consultation meetings in the different countries, with civil society, with governments, with regulators, and with ethics committees, as well as through setting up our global community advisory groups. And we think that this really helped us create the type of trial and study it in the sites, the locations, and the participants who are most disproportionately affected by HIV, which would help us eventually implement lenacapavir when it's approved and available and globally. So I'll jump into the results now. So many of you have seen this study design slide. This one's a slightly updated one because I like to emphasize something that may have been lost in our explanations of it in the beginning. And as you all know, we did a cross-sectional incidence cohort to assess the background HIV incidence, and that was done with recency assays and the RITA testing algorithm. And the primary endpoint was comparing incidence on the investigational product, either lenacapavir or FTAF, and comparing it to background HIV incidence as estimated by the cross-sectional incidence cohort. But there's other couple of things that I want to highlight with the study design. Every single person was loaded at day 1 and day 2 with 2 pills of lenacapavir if they were in the active arm on day 1 and day 2, in addition to their sub-Q injections. Now, if they were in the oral arms, they got len placebo pills. And that's critical to know because we achieve the target len concentrations 2 hours after the day 2 oral dosing. So it's really critical as we think about ways to implement the drug that people understand the importance of day 1 and day 2 loading. It's really to boost your PK so you are protected by day 2. Similarly, in the Purpose 2 study design, this included lenacapavir and Truvada, in contrast to Purpose 1, which also looked at FTAF in cisgender women, in adolescent girls, and young women in South Africa and Uganda. Purpose 2 took place in 7 countries. So South Africa, United States, Brazil, Peru, Mexico, Argentina, and Thailand. And one of the things that was critical about this study is we heard from a lot of people about the importance of inclusion of trans men and gender non-binary people who had not been specifically and intentionally included in in pivotal trials. So this trial included cisgender gay men, trans women, trans men, gender non-binary people who had sex with participants assigned male at birth. So truly a diverse, inclusive study. And this study also included the appropriate dosing regimen with the day 1, day 2 dosing. And again, target LEN concentrations are achieved 2 hours after the day 2 oral dose. So as previously discussed, this was a geographic distribution, and you can see the distribution of the participants. And we think that this pivotal trial registrational program is really the most globally, racially, ethnically, age, and gender diverse phase 3 program conducted to date. Excuse me. Today is the first day I'm really talking because I lost my voice yesterday, so I apologize for having to take a water break. Anyway, you can see here that 67% of the participants were non-white, 63% of Hispanic or Latin ancestry, and 22% gender diverse in the pivotal trial. So here are the baseline characteristics. Just want to highlight a few things here because many people have seen these results before. But to know, Purpose 1, of course, went from 16 to 25, and we did have a nice chunk of 16 to 18-year-olds, about 124, which is about 3 times the average amount of adolescents in a dedicated adolescent study or pediatric study. In Purpose 2, the age range of 16 to 25-year-olds was about 1/3 of the study. And just to note that the max on the age range went up to 74 in Purpose 2. So really a diverse range of 16 to 74-year-olds. And other things to notice were the prevalence of STIs and screening. So sexually active population with lots of STIs, as well as in Purpose 2 reported chemsex. And, approximately 23% had prior lifetime use of PrEP in Purpose 2 and about 6.5% in Purpose 1. The vast majority of Purpose 1 and 2 participants had never used PrEP previously, but we did have a chunk that had previously used PrEP in the Purpose 2 population due to the geographic distribution and age of those participants. Okay, so the results, people have seen these results. We had 0 infections in lenacapavir in Purpose 1 and only 2 in Purpose 2. This was associated with 100% efficacy of lenacapavir compared to background HIV incidence and a 96% efficacy compared to background HIV incidence in Purpose 1 and 2, respectively. With respect to FTAF, the incidence on FTAF was not different from the background HIV incidence. With respect to superiority analyses compared to Truvada, Len was superior to Truvada by 100%. And 89% in Purpose 1 and 89% in Purpose 2. Of no, FTAF and FTDF were not numerically different in terms of incidence. So we wanted to explore the Len and FTAF results in Purpose 1. So here, what you're seeing in the blue box is the injection on time adherence. And you can see injections were on time for over 90% at weeks 26 and week 52. And the on-time injection rate was similar for Len and the placebo injections. In the graph, you're seeing the classic way we present our DBS results, dried blood spot analyses. So green is 4 or more pills per week. Yellow is 2 to 3 doses per week. And low or red is less than 2 doses per week. And you can see even at week 8, only approximately 22% of the people were taking 4 doses or more per week. And that the green and yellow really declined over time, or another way to look at it is by week 52, 93% of the people in this representative subsample of the study were taking less than 2 doses per week. With respect to the Purpose 2 results, the Len injection on time percentages were very similar, of above 90% at weeks 26 and 52. And the on-time injection rate, again, was similar between Len and placebo injections. But here, in contrast, you can see a real difference in the adherence, both to start off with and in terms of persistence of adherence. So more people in Purpose 2 in the representative DBS cohort were taking their Truvada at 4 or even 2 or 3 doses more per week. Just to remind us, FTAF and Truvada are both highly effective for HIV prevention, as I'm pretty sure everybody on this call knows. But I just wanted to highlight the the dissimilarity of the graphs from the Discover trial. And I think this demonstrates something that we all know since the 13 plus years from the results of the original oral PrEP trials, which is oral PrEP, it works when taken. And in trials with high adherence, like in Discover, you can see high efficacy of the FTAF and Truvada arms. And so to better understand the results of the FTAF data in Purpose 1, we did a matched case control trial, which is pretty classic and has been done in most oral PrEP trials. So here we show on the left-hand side, again, the green, yellow, red breakdown in the people who acquired HIV compared to, on the bar on the right, match controls. And you can see far more people who acquired HIV had low adherence. And the odds of HIV acquisition were 89% lower in women who took at least 2 or more pills per week. And on the right-hand side, I just have the Discover results to show you that in contrast, in Discover, you can see the match controls had a much higher percentage of adherence. But again, most of the people who acquired HIV in the Discover study also had 2 pills or less adherence. So similar results in terms of who acquired HIV, but very different results in the matched controls. Okay, so in Purpose 2, we get a lot of questions about why these 2 participants seroconverted on LEN. And I just wanted to highlight a few things about that. First, there's only 2 in over 3,300 people in this trial all around the world. So we're not going to give you more details than we've revealed in the very detailed Purpose 2 New England Journal paper, what I'm going to say here today to protect the privacy and confidentiality of these 2 participants. But what we can say here is in terms of Purpose 1 and Purpose 2 testing, so people can understand, all participants had point of care HIV antigen antibody testing and central antigen antibody testing at all scheduled visits. And participants did have viral RNA load testing at baseline. For both participant A and participant B, they had were diagnosed with HIV with standard HIV serologic testing at weeks 13 and 26, respectively. Neither of the participants had any evidence of delayed HIV diagnosis by RNA, including single copy RNA testing, and they both had LEN concentrations that were within the range of the random 10% subsample of LEN participants and in the range of LEN concentrations in prior studies. We also, I briefly alluded to this, that we included participants who were not required to take contraception in Purpose 1. If they wanted to take contraception, it was freely provided to them. But a lot of people wanted to get pregnant, and they did. And we had several pregnancies in lenacapavir, FTAF, and Truvada. The rate of the expected spontaneous miscarriages was very similar to the rates observed in these geographies. So the available pregnancy outcomes were very similar to that of the overall population. Both in both trials, lenacapavir and FTAF and Truvada were safe and well tolerated with AE profiles consistent with prior trials. There was a slight difference in eGFR in Purpose 2 between the LEN and Truvada arms, which may have been related to higher adherence to Truvada in that trial, and the age range of folks. Okay, so as we all know, LEN works by forming a drug depot. It is administered under the skin, in the fat layer under the skin. This drug depot may be palpable in some people. It's usually not visible in rare instances, and in almost all the instances where there is a significant ISR, it's related to the technique of administration. As the drug elutes out over time, the depot gets smaller and the drug goes into the plasma to protect the participant, and the nodules can resolve and reduce substantially in size prior to the next injection. We found over the course of the study that counseling on the mechanism of injection, giving people ice, and injecting at the correct angle and doing post-injection ice significantly improved people's outcomes, and that's shown in these slides. Green here is no injection site reaction, and you see the most common injection site reactions reported in PURPOSE 1 and PURPOSE 2. So overall, based on the green, most people did not have any injection site reactions. Most folks only had grade 1 or grade 2 if they did have them, and they declined over time, which has also been noted in other injectable trials, including other trials of lenacapavir. And in total, the injections were incredibly well tolerated. With over 95% of participants choosing to continue on to the open label extension. And in the open label extension, participants can receive lenacapavir in the abdomen, upper arms, upper buttocks, and thigh, obviously subcutaneously, and they'll continue to receive len until it's available in their location. We filed in the US, EU, and with EU Med4All, South Africa, and Brazil, and the team is incredibly busy doing all the rest of the global filings this year. And we've been granted priority review with the FDA, so we expect lenacapavir approval on Juneteenth. And we're granted accelerated assessment with the EMA and EU Med4All, so we expect those approvals later this year. And this was a big year for subcutaneous twice yearly lenacapavir, but we didn't stop there. And I just wanted to highlight our once yearly data that we showed at CROI. So we have 2 different intramuscular lenacapavir formulations of the free acid rather than the sodium salt, which are lasting quite a bit longer than 52 weeks with the current doses that were administered in this phase 1 study. And the concentrations for both of those formulations, is shown in green and blue, were higher than the twice yearly len concentrations. And so we are ready to move forward with the phase 3 clinical trials later this year. And I just wanted to highlight, we have lots of anecdotal data in PURPOSE 1 and 2 about ICE, but in this study, we actually did do ICE in one cohort and show that more people more people who received ICE had less pain. Maybe that's not the most straightforward way to say it, but ICE definitely seemed to reduce the injection site pain, which wasn't terribly common to start off with in these 2 formulations, as you can see here by most of the blue and green on the slides. And then we're continuing on our work and also looking forward to long-acting orals. And you know that we're evaluating different long-acting orals for treatment, and we're also looking at their potential for weekly and monthly dosing in HIV prevention. So thank you very much, and I'll
Thank
stop
you.
sharing
Great.
my slides.
Over to you, Rebecca.
Thank you. Okay, now you're seeing my slides. I'm going to switch to presenter mode. Just want to make sure that it's
Yep.
looking okay.
They look good.
Okay, that's okay.
No, now it is.
Okay, now it's good. All right, and there's nothing's blocking my screens.
All good.
I'm seeing something here that says I'm sharing my screen. Whoops. Okay. All right. So first of all, I really want to say the work that's been done so far is amazing. We have a lot of great PrEP options, either approved or soon to be on the market and in development. It's really encouraging. So as we've heard about already, the oral daily tablets that work for many people, and then the Cube 2-month or 6 months a year injectable that Maggie talked about, and even less frequently coming up. And Mopali talked about the Q6 months and possibly even Q12 months injectable. And that's great. And we still think that there is a space for an oral monthly tablet, which is 8527. So this is currently in phase 2. It has the potential to contribute to the field. We think that people can benefit from, improved discretion, right? Not everybody wants to either store a bottle with 30 tablets in it. Some people also don't want to get injections. maybe not having the infrastructure in place to be able to have injections or go the last mile. And some people just don't want to get a shot if they can help it. But I want to recognize that it's just going to be different for different people. It's going to be different for different health systems and different providers. And I just want to use the example of the uptakes of contraceptive methods, that the addition of one method available to at least half the population correlates with an increase of 48% in total usage. So like we talked about a little bit earlier, increasing the pie, like this is also going to increase the pie if we can give people options and they can switch back and forth, et cetera. So we think that this really has an important place to contribute to the future of PrEP. And just to take a step back for people who have not been following the story about the nucleoside reverse transcriptase translocation inhibitors, the NRTTIs. So a nucleoside analog, the way that they have been working until now is that they are obligate chain terminators, right? They're missing the 3' hydroxyl group, which obligates the growing DNA chain to terminate because it just can't grow once the nucleoside analog has been incorporated into the growing chain. The difference with the nucleoside reverse transcriptase translocation inhibitors, even though they are nucleoside analogs, they keep the 3' hydroxyl group, and then they also have a 4' ethynyl group, and I've laid that out there for people. And one of the reasons that this is important is because these groups actually make the NRTTI bind very tightly into the active site of the enzyme, and so then it doesn't want to move, right? It just gets stuck in the active site, and so then the chain can't elongate because there's no active site for the next nucleoside to move into. And if for some reason it does move out of the active site and another nucleoside is incorporated at that 3' hydroxyl group, there's a conformational change that's induced by the NRTTI being in the chain, and it moves out of the active site and the chain is terminated anyway. So it works by more than one mechanism, but I wanted to point out that it is unique that it does have that 3' hydroxyl group. I'm just going to start off by giving people an overview of our non-human primate model. This was presented last year at the R4P conference in Lima. This was a rectal challenge PrEP model in macaques, and there were 8 macaques per group. And what happened is that every week they got a dose of 8527, and then 10 weeks at the same kind of cadence, they got SHIV intrarectal challenges, and then plasma 8527 was measured, the triphosphate was measured in PBMCs and in rectal biopsies, and then uninfected animals could be re-challenged. And I will preempt the question about why we're looking at weekly doses here, but in humans, we're talking about monthly doses. It has to do with the metabolism is just much faster in macaques. And so we can't do monthly dosing in macaques that would replicate what something would look like in humans. So this is as all non-human primate studies are there. It's a model, but it's not exactly the same as what we're looking at in humans. So here's the infection curve for this study. And I'm only showing the lowest dose that was effective, which was the 0.1 milligrams per kilogram dose, but we started with a much higher dose. So we started with 6 milligrams per kilogram, and then in a stepwise fashion went down to 2 milligrams per kilogram, 1 milligram per kilogram, 0.3, and then finally 0.1. There were no infections in any of the animals that received MK-8527. And the reason you see kind of 2 control lines there is because we wanted to make sure that there wasn't something about the way that 8527 was being delivered that was preventing these infections. So the first line there is where macaques didn't get any intervention, and that was the control arm, and you can see the rate of infection in those animals. And then the dotted line, they actually repeated the kind of sequence, and they gave the macaques the vehicle in which 8527 had been prepared. So basically it's still no intervention arm, but we just wanted to make sure that there wasn't something about the intervention itself that was contributing to the really effective levels that we saw with 0.1 milligrams per kilogram. And here are the triphosphate concentrations that we think the dotted line there would be the effective dose that we think would be for prevention. And you can see the bottom scale there is the 0.1 milligram per kilogram dose, and how it is is the stepwise up to 6 milligrams per kilogram, but we stay above the doses that we think would be effective for prevention in humans. It correlates with the dose in humans. So here is a study that was presented at CROI in 2024. These were the single doses given to participants who are living with HIV but are treatment naive. So these doses were as low as 0.25 milligrams times 1. That is the kind of orange line there, but that did not achieve the decrease of at least 1 log drop in the viral load that we expected to see or that we wanted to see to determine efficacy. So the lowest dose that reached the threshold we had predetermined was 0.5 milligrams. And again, this is a single dose. And I'll just say that the PK was similar between people who are living with HIV and those who are not living with HIV. So again, after a single dose of 0.5 milligrams in humans, to 10 days, you can see that the viral load decrease is quite substantial. And then this is another study that was presented at CROI in 2024. This is the single ascending dose study and a multiple ascending dose study. What you're seeing on the left-hand side here is a 1 week period after the first dose. And then we skip the second week. And then the next panel on the right, what you're seeing is the longer follow-up that occurred in people who got that third dose. So it's the first week, and then the second week you're not seeing, and then it's the third week. And these doses were 5 milligrams once a week, 10 milligrams, 20 or 40, all once a week. And the dotted line there is, again, the threshold that we want to be above for protection, that we think will be protective. And the half-life here of the triphosphate, which is the active form, is about 10 days, 9 to 12 days. These were really well tolerated. And you can see that even with 10 milligrams per week, we're staying well above the threshold that we want, even out to 28 days later. So the phase 2 study was conducted recently in individuals who are at low risk of HIV exposure. This was a randomized trial of 2 to 2 to 2 to 1, people getting randomized to MK-8527, either 3 milligrams, 6 milligrams, or 12 milligrams compared to placebo. I want to say the reason why these people had to be at low risk of exposure is because there was this placebo arm. So we didn't want people enrolling thinking that they would be getting necessarily protected against HIV. And the reason we had the placebo arm was, of course, to get a very robust sense of the safety and tolerability of 8527. So these people also could not be pregnant or breastfeeding. And I want to just call your attention to the fact that there is no risk benefit calculation to be made here because people were not intended to be benefiting for this study. So we did not want people either being pregnant or breastfeeding in this study. And we didn't want them having prior use of islatravir or 8527. We wanted people to have a fresh look and not just say, oh, when I took islatravir, I felt this or that. So they had to be essentially naive to both of those. And so there were 6 monthly doses and then an 8-week follow-up in a blinded fashion. So safety, tolerability, PK, we had some enrollment targets that we met. It was conducted in the US, Israel, and South Africa. And the doses that were selected were presented this year at CROI in a poster. And you can see the clinicaltrials.gov number there if you want to learn more about the study. I'm also happy to take questions, of course. So in summary of what we know so far about 8527 as oral monthly PrEP. It's a novel oral nucleoside reverse transcriptase translocation inhibitor that's phosphorylated intracellularly to the potent and specific inhibitor of HIV-1 in treatment-naive people living with HIV. Single doses as low as 0.5 milligrams achieved more than 1 log drop in viral load at day 7 after the administration, has a very long half-life with about a 10-day half-life of the triphosphate, which is again the active form, and it's been well tolerated in phase 1 and phase 2 studies with no clear dose-related AEs. And I will say that we hope to present the full results of Protocol 7, which is the phase 2 study, at a conference later this year. And so I'll say thank you, and I think we can, I'll turn it back over to Patrick, I think.
Thanks a lot, Rebecca. Actually, I'm going to moderate the Q&A. So If people have questions, you can raise your hands. You can put other information in the chat, but this was really wonderful. So thank all of you. Carolyn Mullis had a question, which was very much on my mind and very much on other people's minds, which is in the wake of the current administration's curtailment of a lot of the research that may inform the better deployment of these products, what are each of your companies planning to do to help support scholarship so that we can learn as much as possible in the rollout of these exciting modalities?
So,
Go ahead.
okay, I put 2 links in this chat specifically for early investigators. So there's the Gilead Research Scholars Program, as well as the Research Scholars Program specifically for public health that support junior investigators. And more broadly, as Maggie highlighted, you know, it's critical to do implementation science no matter how big or diverse your pivotal trial program is. Clinical trials are different than the real world. So we are, you know, launching our implementation science agenda this year, and we'll be putting out requests for proposals for implementation science with lenacapavir, as well as doing collaborative studies similar to the prior efforts for approved PrEP products to better understand the use of lenacapavir in real world settings. So those will be opportunities. And I will reiterate my CEO's answer to this question because he's the one who would be answering it. But we are working tirelessly to do whatever we can to influence and support the agenda for science and infectious diseases and HIV prevention and, you know, scientific advancement and drug development through all the means that we have. And so it's going to take everyone to move the agenda forward, and we're trying to do everything we can. And I know our industry organizations are also working on that collaboratively across biotech and biopharma.
Maggie, you were going to say something?
Yeah, I mean, I think that was well said. I think, so first of all, I am so sorry to the entire community. I mean, what is happening currently in the HIV landscape, both on the treatment and prevention side, is terrible. And I think we all acknowledge that. And it's a huge tragedy for all of us, for the community. So, and we're appreciative of that. And we're monitoring the situation for sure. There is a lot of strategic thought going into how we can plug some of those gaps. I mean, obviously, I don't think we can plug everything. The devastation is quite vast, and new holes are being drilled in our programs every day. But there will be opportunities. We're continuing our investigator-sponsored program. There'll be future RFPs. We're working with our current studies to make sure that they are supported and ongoing as much as we possibly can. I think in the meantime, you know, I think it is kind of imperative for us to definitely share the knowledge that we have so far, because as much as we want to continue to support our research portfolio, we want to support the patients as best as possible. And we do have some great data, at least that we've already started to generate around best practices for long-acting PrEP, and sharing those best practices as we help to kind of continue this collective effort amongst the HIV community is really important. I shared with you that we do have kind of a diverse portfolio answering lots of different questions. And I encourage us all to kind of collaborate and figure out what we can do in the meantime to best serve our patients as we start to build up this research portfolio again. But there will be opportunities. Please reach out. Conversations are ongoing. We are 100% dedicated to HIV, and we'll continue to support research moving forward.
Hey, Maggie. Rebecca?
Yeah, I share the sentiments of many people on this call, and also the sentiments of my colleagues. I think I speak for everybody when we say that we feel like we're part of this community too, and our intention is to support public health. our program is a little bit of a different situation, right? Because we are yet to start our efficacy trials, and that's going to be a different way to continue to support investigators by partnering for the next steps of the investigations around 8527. I'll say that Merck also has a big treatment group, and we are committed to continuing to develop also medications for treatment. And just like Gilead and ViiV, we also have an investigator initiated program, and we do support grants that for people applying directly to us. And I can put the link to do that in the chat. But we're also very committed to maintaining momentum in HIV treatment and prevention. And like Maggie said, we can't fill the entire gap, but we're doing what we can.
It's Rebecca. Alberto, I don't have the ability to see if people have their hands raised. The screen right now is just the 6 panelists. I don't know.
Yeah, I don't see any raised hands.
Okay, thank you.
Yeah.
But I don't know if people can go off mute, because Julia Marcus left a comment in the chat, and your comments all dealt with some of that, but I don't know, Julia, if you have any additional issues you'd like to raise with the panelists. And thanks. So Julia thought the responses were helpful. I do think that there is a sort of parallel question, which is more than just the support for future clinical researchers, the actual support for programming. Are there any conversations that you're having internally about how to ensure access for these wonderful new modalities, given some of the changes in sources of public funding for HIV services as well as research?
We're definitely looking into it. We're kind of considering all options. It wasn't something I think any of us were prepared to do, until just recently. So it is a pivot for us as well, and to figure out how to kind of divert funding or change funding mechanisms is not something that happens overnight either. So we're trying to figure out how to best support the community to be able to deliver the medicines to the people who can benefit most. So more to come. I don't have something specific to share, but it's definitely something that's top of mind. And I feel like a part of every conversation that we have at ViiV on a daily basis. So
great to know that's happening. Thanks.
Yeah, absolutely. I mean, as you know, last year we announced our voluntary licensing agreements with 6 generic companies for 120 countries. We're continuing to work on figuring out manufacturing and other options for lenacapavir globally, so it can get to all the locations where people are disproportionately affected by HIV. We're continuing our global filings. We are partnering with whomever with whom we can partner, directly with governments, with global organizations that are not impacted by the loss in US federal funding. But many of those institutions, as you can imagine, are getting tremendous calls to make up the deficit. And it is a challenging time, but we remain committed, and we are, if you want to think about, is a company committed or not, think about what they're doing with how much supply they're making. And we're making all the supply to get it to where it needs to go. And none of that has stopped or slowed down, because that's what we want to do. Like all of us have been, all the people on this call have been working on HIV since the beginning of our careers, and we are all absolutely committed to making sure, like me, my team, our colleagues and partners, that lenacapavir gets to where it needs to be. In some ways, long-acting options have a benefit over daily oral pills in terms of adherence and persistence over time, which we've already seen that could be important in a time like this. But there's, pros and cons and challenges, of course, but it's something that's really had a tremendous impact on us as we think about how to file and how to get global access. We've already done several innovative things with access in limited settings throughout, and we're continuing to do that, like what we did with remdesivir during the pandemic. And we're working with all of those partners as well. So I'm sorry I can't say anything concrete. There's no magic bullet. It's a epic cluster something. But we're all working on trying to figure it out. That's the technical term I've been using.
Great. Well, thank you. I know for Rebecca, it's early days. I don't know if you have any
Yeah.
response.
Well, I'm going to say we are learning a lot from the experience from ViiV and Gilead, and Merck has also experience in voluntary license and things like that from the also from the COVID pandemic, molnupiravir. So we're building on that experience, and we're watching very closely and trying to follow the good example from our colleagues.
Great, thank you. Julia, any other comments you have? You're unmuted, and then we'll go to David Hardy.
No, that was really helpful. I think I think because of where we are right now, it's it's front of mind. And so I think hearing that you all are aware and doing your best, and of course, like as you said, this isn't something you've thought about before, and we're all in a new situation, but just great to hear that there's attention to ways that industry can step in. And of course, you can't fill the gap. There's no way to fill the gap that's been left. But yeah, that was just very helpful. Thank you.
Thanks, Julia. David, you want to answer your question? You can unmute.
Hi, this is David Hardy from Los Angeles. I just wanted to say thank you, first of all three of you, for updating us on your product development programs. It's all really great to see. My question is not so much about implementation, but so much about rollout and actually getting your drugs into the patients. You know, as ViiV has noticed, the uptake of cabotegravir for PrEP is slow. It has had very small market share. It has been difficult for many, certainly private practitioners, to do it. A little less so for many publicly funded clinics, interestingly. But my question is, what is your company doing in addition to producing great science and clinical trials and implementation studies to actually practically get the drug out there, to work with the payers, to work with the pharmacy benefit managers, who seem to be the biggest hindrance in terms of actually getting the drugs to the people? In terms of ViiV, what are you doing now? And Gilead, what are you planning?
Yeah, so no, thank you for your question, Dr. Hardy. I mean, it has been a challenge, right? Like I will say CAB, long acting for both treatment and PrEP, has not it has had many challenges along the way. First, it was COVID. We were kind of rolling out HCP administered medication during a worldwide pandemic where a lot of people were not leaving their house, let alone going to their doctor to receive care on a regular basis. And now with all these government challenges as well. The insurance challenges have been another barrier as well. But what I can say is that we have been able to start to develop a lot of these implementation strategies and support for benefits verification and inventory management that are really helpful to clinics. It took us, there is some learning curve here. It's different than delivering oral medication for sure, and that it's a new kind of skill for clinics and clinicians and staff to learn. But now, coming out of the implementation science studies and kind of real world experience, we're learning kind of what the best practices are. So moving forward, there's a number of best practice sharing models that we're going to be rolling out moving forward, because there are clinics who have started to figure it out and who are doing quite well with it. And it's identifying those models and sharing those best practices more widely that is going to help us kind of accelerate even more. We've done better than, if you look at some of the AVAC data that they've published, we've done better than what we've done with oral PrEP, but we want to do even better moving forward. There's a number of digital tools available, ViiV Connect, to help with acquisition and view to help with inventory management in clinic that can be very helpful to people moving forward. So I highly encourage, I'll put those links in the chat, highly encourage those people, those of you that haven't accessed those tools. Those are really beneficial in getting clinics on board, as well as a number of resources on our website. I'll put the links in the chat as well for you to learn from. But I do think best practice sharing is kind of key here, because there are really great strategies that have worked. Now, I practice in the VA healthcare system. My history is practicing in the VA healthcare system. We have 168 clinics. And it's crazy to me still that even in an integrated healthcare system like that, there are clinics that have figured it out, and there are others who haven't. And even if clinics are very similar, we need to share those best practices. So if you have found strategies that work for you, I highly encourage you to share that information with others, especially when it comes to benefits verification and specialty pharmacy management. It is something new. There is a learning curve to it, but people have overcome it, and we need to kind of work together to get it to the point where we can get the medication to patients as easy as possible.
Thanks. I'll just say that we have benefited tremendously by watching the rollout of CAB and trying to learn from our investigators and colleagues and partners who are implementing CAB, and really trying to understand what has gone well, what is relevant to lenacapavir rollout. And this is something teams at Gilead, people way smarter than me who understand and have like MBAs and understand finance and all that stuff that I don't get, have been working on for years, since we launched the trials, to try to figure out the best way to make sure people have a smooth and enjoyable and straightforward experience in ordering, administering lenacapavir, and receiving lenacapavir once it's approved. We've done a lot of things differently in this program. Like, Gilead helped fund and initiate the medicines patent pool way back in the day. We were absolutely critical in helping set that up for ART globally and for many of our other medicines. And we continue to think about the best way to get things globally. And, it's kind of a mix in this conversation about talking about getting access in the United States versus getting access globally. But we have announced our voluntary licensing agreements even before approval anywhere. Immediately after the results of Purpose 2, and we've been working on those agreements for a long time in anticipation of positive results. So there's lots of things that we've started a lot earlier than we normally do. We're having discussions about implementation way in advance of when we do. And we were, in fact, quite surprised by both the trials reading out early, but we were planning for the best and, making sure that we had these plans in place. So we are fully committed to trying to make sure Len gets to the people who need it the most.
Thank you. And it's early days, Rebecca. Any comments you have?
I don't have anything to add. Yeah, we're watching and learning.
Absolutely.
Yeah.
So a question from Connie Kellum. What are the implications of the new formulations for the Q4 month cap and Q year Len in terms of generic production, the agreements, licensure timelines, etc.?
I mean, it's very early, Connie. You're assuming,
Yeah.
I mean, we have to do the pivotal phase 3 trial. Now, I mean, like we're at like 2019 with 12 month, as where we were with 6 month. But obviously, we're going to pursue a similar approach.
Yeah, same. I think it's still early days, but we also want to look at an expedited process. Now that we've been through the process, we want to make sure that it gets better and better with each of the newer long-actings that come forward.
Well, let me ask some more concrete questions about the here and now. In terms of RNA testing, what do you think the role of RNA monitoring is for long-acting medications at this stage, given what we've learned recently from Purpose 1 and 2 and some of the follow-up work on CAB. Do we really need to be doing as much RNA monitoring as some of the guidelines might suggest?
I'll let Maggie take that for CAB, and then I'll answer for Len. Because I think it's different.
Yeah. So I think what we're seeing is that also in the prioritization of resources globally for HIV, we need to do what's best for our patients. And so in resource limited settings, I think there is an argument to be made about streamlining testing requirements to do what's best for our patients. As I had shown in the real world data cohorts, we're seeing 0 new infections in the CAB-LA arm, which is very reassuring in the real world data that we're having no infections moving forward. Right now, according to the label, we do require the HIV RNA testing, and that's what we need to do in the US. In our PILLAR study, and we just presented these results actually at CROI. So in our PILLAR study, we told them that they needed to follow the label, but we watched and allowed the clinics, we monitored the clinics and to see what kind of HIV testing was done routinely with their patients. And what we saw was that there was a little bit of a mix. So the clinics that had no experience with PrEP prior were more apt to do both RNA testing and antigen antibody testing at the same time, whereas people who had clinical experience with PrEP were more comfortable with just doing antigen antibody testing moving forward. So more to come as more data kind of rolls out, but I think simplifying the HIV testing algorithm would be a benefit to everyone, especially our patients.
Cool. And there are real world data from OPERA and TRIO as well that show that a lot of clinicians aren't doing the RNA testing. So it's just a question about, if we can make things less onerous and less expensive. So
exactly. Yeah.
Yeah. In the Purpose 1 and 2 studies, we only had 2 infections, but neither of those infections would have been recognized earlier with RNA testing, and neither of those infections were baseline RNA, were baseline infections, right? So they were diagnosed after. I think, we'll have to see what the US and European labels say. They're being evaluated by the regulatory agencies, and obviously it will be a discussion. Our hope is to have a more streamlined process because we understand the implications of RNA testing for cost. That being said, in our implementation science studies that we are collaborating with, this is an important question. So we're thinking of the best way to study this in low and low middle income settings, so we can get a definitive answer for those settings, which I think will be really helpful. And I think it's a perfect implementation science question. So we're working with many of our collaborators and experts in implementation science to best answer that question.
Connie has a question, which I actually think is, she's asking about Len Q12, but I think it's actually relevant also for CAB Q4, and brings us towards the end of our time together. A good study design question, which is, what would the phase 3 studies look like for these longest acting modalities now for your respective compounds for Q4 CAB and Q annual? Len, how does one show efficacy in an era where we have such highly effective modalities that are already pretty long acting?
Do you want to go first, or do you want me? No. I think right now we've just launched our Extend 4M study, which is our 3 times a year CAB ULA study. It's a PK bridging study, phase 2 currently, to look at PK profile. And then we haven't settled on a phase 3 study design just yet, but I agree. now that we have the data, how can we be most effective moving forward?
We haven't fully fleshed out our design, so I can't really comment on the designer timeline at this point, but be happy to come back and talk to you all about it later.
This group, I'm sure, will be very interested in, and Connie's aware, as you all are, but maybe not everybody on the call, that the Forum for Collaborative HIV Research has sort of a working group that is well represented by investigators in industry, and that's really trying to grapple with these questions. What do you use for equivalence, or what kind of counterfactual can you use to, as we start getting even longer acting and even more effective modalities. Very hard to design studies, but I think we've seen with Discover that it can be done. So just have to keep moving forward. I'd like to give our panelists a few minutes to close, make any kind of closing comments that you'd like to make. Maggie, anything you'd like to add on the discussion today?
Just to say thank you all for your time. I know right now the future of HIV and HIV prevention does not feel very optimistic with all the changes going on in the US government, but I really am still hopeful. We have these great new medicines that are being generated from all of these companies that really can benefit patients. And it's up to us to kind of still band together as a community and learn from each other and really support the HIV advancement of science to be able to get to the end of the epidemic. I am still hopeful that we can get there, even though we've had challenges along the way. We've always had challenges along the way. This is a very resilient group of people and a resilient community. I know that we may feel a little deflated, but hopefully we're not defeated, and that we can come together and do better in the future. So thank you for your time today.
Mopley.
I don't really have much to add other than Maggie, like you channeled everything that I was going to say. I guess the only thing I'll add is, you guys know I have 8-year-old twin boys, and we watched Finding Dory recently, and I feel like the mantra from Finding Dory is incredibly helpful right now, which is just keep swimming. And just one fin in front of another, if that's appropriate for swimming. But we just have to keep on doing what we can and trying to move forward. And I also tremendously believe in the resilience. I remember the last time when I was working in public health and we got a list of words that we couldn't say in grants. And that was like over 12 or 13 years ago. And like we motored through that. And I'm hopeful that we will motor through this as well. So just keep swimming.
Yeah, and I'll just say, I want to say thank you for having us and listening to what we have to say about these new products, and recognizing that new products are not going to be great as they could be unless people have access to them. So everybody's committed, and we're all in this together. And I'd say the community not only is resilient, but also really creative. And so I think that we will find a way to keep swimming. So thanks, everybody.
Thank you. I wish we had like a soundtrack we could play. Donna Summer, I Will Survive. Arlene Stritch, I'm still here, but really want to thank our fabulous panelists on behalf of Patrick, Sullivan, and myself, and thank you all for participating. Really very thoughtful and helpful discussion today. Be glad to invite you back in the future too. Thank you.
Great. Thank you so much.
Thank you.
Thanks, everyone.
Bye.
Really appreciate it. Yeah.